Go ahead, ma.
So, good morning, everyone, all the participants online and offline, and Dr. Jagadis Gupta, sir. Thank you. Thank you so much, sir. We are glad to have you here. So, for the benefit of all, I'll read about his biodata and briefly regarding the work. I hope I am audible online also.
Can anyone confirm whether we are audible from here?
Very well.
Yeah,
thank you.
Thank you. Thank you. So, Dr. Jagadis Gupta Kapuganti, he's a staff scientist 6 at the National Institute for Plant Genome Research in NIPGR, New Delhi. He joined this institute in 2014. He has a PhD from University of Würzburg, Germany, postdoctoral fellow from Max Planck Institute of Molecular Plant Physiology, Germany. He was a staff at University of Rostock Institute in Germany. He's a Marie Curie Fellow and worked at the University of Oxford, UK, and as well as he's an academic visitor for the same. He's a visiting Max Planck Institute in Germany, and he has a DST DAT Fellow, Germany. He has prestigious fellowships, to name a few, or a major fellowships are Fellow of National Academy of Sciences, Fellow of National Academy of Agricultural Sciences, Fellow Association of Biotechnology and Pharmacy. He has earned several awards and accolades in his career, to name a few. The Birac Big Ignition Grant Award, Ramalinga Swamy Fellowship from DBT in 2014, Innovative Young Biotechnology Award, Biotechnologies Award in 2015. Moreover, he was listed in world top 2% of the scientists in the database published by Stanford University. His research work majorly focuses on the functional role of nitric oxide in flooding stress that provides new insights into the mechanisms, metabolic adjustments associated with controlled use of carbohydrate and ATP during hypoxic survival. The another research where his team focuses is a role of nitrogen nutrition on plant pathogen interactions, examining the nitrogen flux in the plant and metabolomic analysis of plant microbial exchanges in the apoplast for better understanding the nitrogen type and the influence on pathogen virulence and plant resistance. Another one, the topic which they are working on in association with the international collaborations is maximizing the nitrogen use efficiency in crops by understanding a nitric oxide mediated modularity switch, wherein non-symbiotic hemoglobin expression, how it regulates the nitric oxide accumulation and modulates the nitrogen channel. So he is an editorial review board of reputed high journal impact factors, and he has several international publications to his credit with citations over 7,000 and H index of 40. So today we are glad to have him here and look forward from hearing you, Dr. Jagdish, on today's topic of nitric oxide signaling and its role in hypoxia tolerance in rice. Thank you.
Yeah. Thank you so much. Am I audible?
Yeah, I think you can just pull down the mic a little bit. Yeah, that'll be fine.
Okay, thank you so much for invitation, Dr. Sundaram.
Can anyone confirm whether Dr. Jagdish is audible?
Yes, sir. Yes.
Okay, Dr. Jagdish, you can go ahead.
Fantastic. Thank you so much, Dr. Sundaram, for giving this opportunity to come here and giving this talk. And also thanks to Dr. Varaprasad also for actually coordinating this. Today I'm going to talk about the role of nitric oxide in increasing the hypoxia tolerance in general and also in rice. Yeah, before going to that, it is very important to know the basic pathways, how actually oxygen is very important for the production of energy. For example, if you take the photosynthetic electron transport chain, the plants use carbon dioxide, water, and energy that leads to production of the carbohydrates. And during this process, they release oxygen. But in contrast to the photosynthesis, during the aerobic respiration, the plants use they oxidize the carbohydrates that leads to production of the carbon dioxide, water, and energy. For example, if you take this pathway here, you see the oxygen is very important for the production of the energy. So, but during the flooding or water logging, what happens is that, so this water forms a layer on the top of the soil that actually blocks the oxygen transport from the atmosphere to the soil. So the roots which are Edenhoffs in the soil, they experience hypoxia or anoxic condition. At the same time, the soil microbes which are present in the soil, they compete for this little amount of the oxygen that is present in the soil. So actually there is a competition occurs between the roots and also microbes for the little amount of oxygen that is available in the soil. Actually, that actually causes, hypoxia condition. Eventually, the roots experience anoxic condition. But what happens is that when there is a transition from the hypoxia state to the reoxygenation, when the hypoxia transient flooding, etc., actually when the hypoxia is gone, the plants experience reoxygenation. So during this reoxygenation, what happens is that reoxygenation, there is an accumulation of reactive oxygen species that actually causes damage to the plants. Okay, so the plants experience hypoxia not only under flooding or waterlogged condition. It should be noted that the plants experience hypoxia during their development. For example, if you take the bulky tissues such as seeds, roots, and stem tissues, you see the oxygen level that is present inside the tissues are very low. So outside layers of the tissues are having exposed to the higher level of oxygen, and but when you look deeper into the tissues, they always experience hypoxia condition. So the thing is that plants experience hypoxia not only during the flooding but also during their development. They actually experience hypoxia. So in our laboratory, actually we are elucidating that in one condition plants are unable to survive, but in another condition, plants are continuously dealing with the low oxygen conditions inside the tissues. And actually, what are the mechanisms actually plants are adapting to survive? The cells are surviving in the internal tissues, and then can we adapt these mechanisms to apply in order to increase the flooding tolerance? So, for example, here oxygen condition profile of the various tissues like pea, soybeans, rapeseed, and roots, and the stem tissues. If you take this one, the oxygen levels are usually actually ranging from 2 to 10 micromoles. That means they are continuously dealing with the low oxygen condition. But under low oxygen condition, what happens is that this is the mitochondrial electron transport chain. So this, when you take this mitochondrial electron transport chain, it has 4 complexes: complex 1, 2, 3, and 4. So the electrons usually come actually and accumulate at a ubiquinol pool, which is in the center of the electron transport chain. Okay, so when oxygen is present, actually the electron transport chain goes in the normal way. That leads to production of the ATP. But when oxygen is actually not present, what happens is that here the electrons started accumulating at the ubiquinol pole, because there is no terminal electron acceptor which can accept these electrons. Actually, that leads to production of ATP. So this excess of electrons accumulated ubiquinol pole, but whenever there is a reoxygenation or low oxygen conditions plant exposed, this oxygen, actually these electrons attack this oxygen, that leads to production of the superoxide. The superoxide that is produced actually leads to a production of the chain of other free radical, because it is a precursor for the other free radical molecule. So what are the negative effects of the low oxygen is that low oxygen stress leads to ATP depletion, because oxygen is not present, therefore ATP is less. An increase in lipid peroxidation occurs because of the excess amount of the reactive oxygen species attack these membranes. Therefore, that leads to enhanced lipid peroxidation, which can damage the cells, and production of the increase in levels of ethanol and lactate, because plants try to produce more and more energy in order to survive. But the byproducts of this fermentation are ethanol and lactate. actually both are actually, if excess amount of ethanol and lactate is produced, that can also damage. And down regulation of oxygen dependent reaction, that means in a cell there are several reactions require oxygen. When oxygen is not present, these processes gets down regulated, and ROS can damage the proteins, and also it causes also delay in the germination process, etc. Okay, so so this hypoxia or anoxic condition usually causes damage to the crops, and annual losses are approximately 40 to 50 billion dollars, and even more they are increasing. So in this regard, actually we looked into a gaseous free radical called nitric oxide, because the nitric oxide production usually actually occurs very high level under low oxygen conditions. So the plants produce this nitric oxide by two different pathways. One is oxidative pathways, second one is reductive pathways. So we actually divided these pathways based on the reaction where oxygen is required. In some cases, oxygen is not required. So oxidative pathways usually occur by a polyamine pathway and also nitric oxide synthase like pathway. But reductive pathways are operational at low oxygen conditions. For example, here nitrate reductase is an enzyme which is present in the cytosol. This enzyme usually what it does is that it converts nitrate to nitrite, and nitrite that is produced is actually converted into ammonia for the amino acid assimilation. But at the same time, this nitrate reductase has a dual role. It has a capability to reduce this nitrite to produce nitric oxide. So thereby, nitrate reductase is an enzyme that actually produce nitric oxide because of its ability in certain conditions such as low oxygen and also pH. For example, if the lactate levels are increasing at low oxygen condition, that also changes the pH, that leads to activation of the nitrate reductase, that leads to production of the nitric oxide. At the same time, here, at the same time, see the nitrate that is accumulated can also be transported to the mitochondrial electron transport chain, where mitochondrial electron transport chain has a capability to reduce this nitrite to produce nitric oxide. So these are the major pathways. One is nitrate reductase and also mitochondrial electron transport chain. These coordinate that leads to production of the nitric oxide at low oxygen conditions. So plants produce nitric oxide, and also plants also have scavenging pathways. So usually there is a homeostasis. The nitric oxide homeostasis usually mediated by the production minus scavenging. So these scavenging pathways are phytoglobin, which is a non-symbiotic hemoglobin, and also S-nitrosoglutathione reductase. These are the two enzymes play very important role in removal of excess of nitric oxide. So the balance of the nitric oxide, that is a physiological level of nitric oxide that is produced, is a production minus scavenging. So then how nitric oxide acts as a signal? It is not a hormone, but how it acts as a signal? That is a question. So nitric oxide mainly acts as a signal by a process called S-nitrosylation. S-nitrosylation is a post-translational modification when nitric oxide reacts with the reduced thiols of the protein, that leads to a modification called S-nitrosylation. So a protein can become active or inactive upon nitrosylation. For example, one of the prominent example here I will tell you is the NPR1 is actually protein. It is in the actually oligomeric form. The S-nitrosylation of this NPR1 actually keeps the oligomeric state. Whenever there is actually stressful condition like pathogen attack, this thioreduction is actually produced. This thioreduction actually breaks these bonds. The nitrosylated NPR1 actually moves to the nucleus, where it actually interacts with the TGA1 transcription factors that leads to induction of the PR genes. This is one of the examples I am giving. A lot of proteins gets activated or inactivated by the process of nitrosylation. Once the protein gets nitrosylated, it can actually play a very important role in the signaling process, such as induction of the genes or activation of the other protein or interact with the other proteins in order to actually carry out the process. So in our laboratory, mainly we are working on the hypoxia-induced nitric oxide, how plants produce nitric oxide at the low oxygen conditions, and then what is this role of this hypoxia-induced nitric oxide in a process such as hyponosty, where there is a PTO elongation takes place, which plays very important role in giving the access of the oxygen to the plants, and oxygen homeostasis mechanism, which I'm going to show, and mitochondrial function, how hypoxia-induced nitric oxide play important role in actually improving the mitochondrial function under low oxygen condition. Oxygen sensing mechanism, where actually there is a pathway called NN-role pathway of the protein degradation, actually which play important role in the stabilization of the ethylene responsive factors, which play very important role in the hypoxia tolerance, and also the production of the arenchyma, such kind of tissues which can help in delivery of the oxygen to the root tissue to improve the survival of the plants. So these are the different pathways we are focusing on, actually elucidating the role of this nitric oxide in various processes that leads to survival of the plants under hypoxia condition. So here, one of the experiments that actually I'm going to show is that here we employed a method called chemiluminescence detection of the nitric oxide. This is a method where you can measure the nitric oxide. What we have done was we have measured nitric oxide under two different conditions. One is normoxic condition, that is 21% oxygen, and anoxic condition, which is 0% oxygen. And then here you see under normoxic condition, if you take this wild type, produce very little amount of nitric oxide. But when we switch to anoxic condition, you see the roots produce a significant amount of nitric oxide. That means anoxia is an inducer for the nitric oxide production. Then what we have done was we took NIA mutant, which is a nitrate reductase mutant. We have actually took and then checked the NO production under two different conditions, same condition. Here you can see under normoxia or anoxia, actually NIA did not produce nitric oxide. This clearly suggesting that nitrate reductase is the enzyme that produces nitric oxide, because NIA mutant was unable to produce nitric oxide under low oxygen condition. But here you see what we have done, we added nitrite NO2 minus to the NIA, and then when you add the nitrite to the NIA, you can see there is a restoration of nitric oxide. This clearly suggesting that, so in the absence of the nitrate reductase, plants use nitrite to produce nitric oxide. That means there is a pathway which is operating independent of nitrate reductase to produce nitric oxide. So then what actually we found that the job is done by the mitochondria. What we have done was we have isolated mitochondria and then supplied nitrite to the mitochondria, and you can see after supply of the nitrite and also reducing equivalent in the form of NADH, here you can see the NO production is induced in the mitochondria. And then what we have done, we used different concentration of the oxygen and nitrogen and checked the nitric oxide production from the mitochondria, and you can see when the oxygen concentration goes down, nitric oxide production is increasing. So that means mitochondria are capable to produce nitric oxide from the nitrite. Okay. Then the question is, why mitochondria is producing nitric oxide in low oxygen conditions in the absence of the oxygen? So in 2003, so there is a Russian scientist, the Vartapetian. Actually, he has done experiments in using rice coleoptiles. What he has done was he checked the mitochondrial structure in the normal condition and 24 hours of anoxia condition. He exposed this rice coleoptiles, and then he found that when you expose to these coleoptiles to the anoxic condition, the mitochondria swell and disintegrate. And then what he found that then he incubated these coleoptiles in the presence of the nitrate NO3, and then he found the mitochondria retained the structure. That means nitrate NO3 has a role in actually protecting the mitochondria when the plant tissue is exposed to the low oxygen condition. But when we thought it is not nitrate, probably it is a nitrate. Then what we have done was we have actually isolated mitochondria from barley and rice, and then we imposed low oxygen, zero oxygen condition to the mitochondria, and then provided NADH and NADH, NADPH to the mitochondria and checked oxidation of this NADH by the mitochondria. And we found that the rice mitochondria without any oxygen, if you provide NADH, they are able to oxidize this NADH. Okay, and what we found that this FCCP actually increases this oxidation, and oligomycin etc. actually reduces the oxidation, and this clearly suggesting that probably the oxidation of the NADH under zero oxygen condition by the mitochondria probably producing the ATP production. Therefore, there is oxidation is occurring even there is no oxygen. Then what we have done, we have actually measured the ATP level in the normal condition and also low oxygen condition in the absence or presence of the nitrate and the nitrite. And you can see here, even without any oxygen, mitochondria are capable to produce ATP without any oxygen, but using nitrite as a terminal electron acceptor. Instead of oxygen, they use nitrite to produce ATP. And then we found anoxic tolerant rice actually has produced more and more ATP than in the barley. This clearly suggesting us rice has a capability, rice mitochondria has an enormous capability to survive, to produce this nitric oxide and produce the anoxic ATP under low oxygen conditions. And then taking this further, what is the impact of this nitrate dependent nitric oxide production on mitochondria? What we have done was we have isolated mitochondria and then imposed them under normal condition and also anoxic condition in the presence or absence of the nitrite. Here you can see in the presence of the nitrite, mitochondria survived much more better. They showed good integrity in comparison to without any nitrite. This clearly suggesting that nitrate has a role in protection of the mitochondria. Nitrate has a role, and in order to actually increase also the membrane potential and production of the reactive oxygen species, and also lipid peroxidation also reduced in the presence of the nitrite. So this clearly suggesting the nitrite products the mitochondrial structure and function. And then also we checked on the role of this nitrate dependent NO production on the mitochondrial structure. So mitochondria has 4 complexes. It is very interesting. The complexes under stressful conditions, these complexes come together and form super complexes. So for example, complex 1 binds to the complex 3, complex 1 binds to complex 4, complex 1, 2, 3 come together also sometimes and form super complexes. Why the super complexes happens is that super complexes increase the electron channeling. Electron has to go from complex 1 to 2, 2 to 3, 3 to 4. It increases the time. So if the super complex formation takes place, this electron channeling, actually the time of electron channeling is reduced. That leads to increase the energy efficiency. So what we found was we have actually isolated mitochondria and checked the complex, these super complexes, and then super complexes actually have enhanced in the presence of the nitrite. That means nitrite can not only help in production of ATP, it also helps in actually increasing the super complex formation that leads to enhanced energy production. And then taking this further, recently, just a few months ago, we published a paper that nitric oxide that is produced in the mitochondria, it helps in mitochondrial biogenesis. Whenever a plant is exposed to stressful condition, when it's not recovering, it recovers enormous amount of energy. And we found that by using mutants and also nitric oxide overproducer, we found that the nitric oxide play important role in the mitochondrial biogenesis. And then later, actually recently what we elucidated was what is the site of this NO production in the mitochondria. And we found that finally, I'm not showing the entire data because it is going to be complex, but we found that the cytochrome C oxidase 6b3 and CoA6L, these are the subunits of the cytochrome C oxidase, play important role in the production of the nitric oxide from the mitochondria. Okay, so then taking this further, actually, how we actually elucidated the role of nitric oxide in hypoxia tolerance is that what we have done was we have actually taken, this is a Arabidopsis system what we have used, wild type hemoglobin overexpression line, which can produce less nitric oxide, NEA12 mutant, which is producing less nitric oxide, and wild type plus CPTIO, which is a scavenger of nitric oxide. So we used this system and grown the plants in the vertical plates, and then we imposed this actually hypoxic condition in the Hypoxia chamber that we have. And what we found that in the presence of the nitric oxide, that means these are all mutants which produce no nitric oxide, but here wild type produce nitric oxide. The plants that produce nitric oxide induced several genes which are involved in primary metabolism and also antioxidant metabolism, etc. And we found one of the protein that is induced is alternative oxidase of the mitochondria that is induced in the presence of the nitric oxide. So then coming to that, then we actually elucidated the role of this alternative oxidase, the protein that is induced under low oxygen condition in anaerobic germination of the rice. So the anaerobic germination is the best example where modulation of the energy metabolism and ROS production takes place. So, for example, if you actually grow the plants, rice plants, we have used a deep water rice here, grow in the normal condition and also low oxygen condition. Here you see what we have done. We have actually grown these rice seedlings in the normal condition and also anoxic condition. We have imposed an anoxic condition, and by using sensor, we have actually measured that the treatment actually causing 0% oxygen. So here you see when we impose the anoxic condition, the germination is several order magnitude higher than in the absence of in the presence of the oxygen. So that means rice has enormous capability to germinate under low oxygen conditions. And then to further actually elucidate the role of nitrite, here you can see we have actually, even in the under anoxic condition, we incubated the we have actually germinated the seedlings in the absence and presence of the nitrate. And you can see here in the presence of the nitrite, there is much more germination than in the absence of the nitrite. That means nitrite has a role in increasing the anoxic germination of the rice. So then what we have done, we have actually checked the enzyme that is responsible for the nitric oxide production under anoxic condition, and we checked the NR activity. Here you can see here NR activity has increased under anaerobic condition, and NR activity has further actually elevated in the presence of the nitrite. And then here, that since the NR can actually produce nitric oxide, what we have done, we have measured the nitric oxide by using a dye called diaminofluorescent diacetate. If anybody is interested, then we can actually share the protocol how to measure nitric oxide by using simple fluorescent microscopy. And here what it does is that this is a dye, diaminofluorescent diacetate, which reacts with the nitric oxide and form DAF2T. If DAF2T is actually nitric oxide reacts with this dye, there is a fluorescence. By using this method, we have measured the nitric oxide under normoxic condition and anaerobic condition. And you can see here in the anaerobic condition, there is an increase in nitric oxide, which is actually correlated with an increase in nitrate reductase activity. And then in the presence of the nitrite, you can see there is a further increase in the nitric oxide. That means when you grow the plants under zero oxygen condition in the presence of the nitrite, there is increase in NR activity, there is increase in NO production that occurs. So it is always recommended, why I'm going to tell is that it's always recommended to measure nitric oxide by using two different methods, because every method has certain advantage, certain disadvantage. Therefore, what we have done, we have actually standardized a method which is called as chemiluminescence detection of the nitric oxide. This is a method which can precisely detect the nitric oxide level. So how this works is that nitric oxide, there is a chamber where nitric oxide reacts with ozone, that leads to production of the highly reactive nitrogen dioxide. This highly reactive nitrogen dioxide jumps from the exciting state to the ground state by emission of the light. The amount of the light that is produced is proportional to amount of nitric oxide. In this method, by this method, even you can measure nitric oxide from the chloroplast, mitochondria, organelles, and very, very low amount of nitric oxide also can be captured by using this method. So by using this method, we have measured again nitric oxide, and we found that our results are correlated with the previous slide I have shown, diaminofluorescent diacetate. So nitric oxide levels have enhanced in the anoxic condition in the presence of the nitrite. So coming to the deep into the mechanism, how rice is able to grow under low oxygen condition. So here, this is a pathway, entire pathway that can actually lead to production of the ATP under low oxygen. So here is the NR that I mentioned. NR can reduce nitrate to the nitrite. The nitrite is actually the can lead to production of the nitric oxide by the NR, and the nitrite that is produced can diffuse us to the mitochondria, and the mitochondria complex 4 is a major producer of nitric oxide, can reduce this nitrate to nitric oxide. The produced nitric oxide again diffuses to the cytosol, and in the cytosol there is a protein called phytoglobin. This phytoglobin can scavenge this nitric oxide. That actually leads to recycling of these protons, that actually leads to the production of the ATP. So this is the pathway that leads to production of the ATP under low oxygen conditions. So then what we found that probably this cycle is actively operating under low oxygen condition in the rice, that leads to production of the ATP. Therefore, what we have done, we have checked the component of this cycle. One is metamoglobin reductase activity, which can also give the indication of the phytoglobin activity. And you can see here under low oxygen condition, the activity of the metamoglobin reductase has increased. And also we checked the phytoglobin, which is a scavenger of the nitric oxide in the cycle, and that the levels of the phytoglobin also enhanced. And also the nitrate reductase 1, NR is encoded by 2 genes, and NIA1 and NIA2. And NIA1 is responsible for 90% of the nitric oxide production, and NIA1 expression has increased under low oxygen. NIA2 expression has decreased. This clearly suggesting that under anoxic condition, when you grow plants under the plants, that leads to operation of this phytoglobin nitric oxide cycle, that leads to production of ATP. Thereby, rice is germinating under low oxygen conditions. And then to further confirm this cycle, what we have done, we generated a promoter reporter constructs of the NIA1 and PGB1. And you can see in aerobic condition, you see these promoters are inducing, even they are further induced in the presence of the nitrite. And then coming to the major part is that what is the evidence that under low oxygen condition, plants are producing ATP? What we have done was we checked the ATP level in the aerobic and anaerobic condition. Here you can see in the presence of the nitrite, you can see there is a enhanced ATP production, enhanced anoxic ATP production. How this ATP is produced is that one is via phytoglobin NO cycle. At the same time, when you take this phytoglobin NO cycle, here you can see this phytoglobin NO cycle. Here the reaction metamoglobin reductase reaction uses NADPH. So this NADPH leads to production of NAD. This NAD is very, very important for the operation of the fermentation in the glycolysis. Okay, so when oxygen levels goes down, there are two enzymes, lactate dehydrogenase and alcohol dehydrogenase. These are the two different enzymes present in the pathway in the glycolysis that leads to production of the ATP under low oxygen condition. So the operation of this ADH and LDH require NAD. The NAD, what we have done was, what we have proven that this cycle also contributes for the enhanced NAD, which can be used during the fermentative process. So then what we have done, we have checked the LDH and ADH activity, alcohol dehydrogenase and the lactate dehydrogenase. You can see the activity of the the LDH and ADH has enhanced under low oxygen condition. At the same time, which correlated with the production of the lactate and the ethanol, these levels of the ethanol and lactate also enhanced in the presence of the nitrite. So, as I mentioned, under low oxygen condition, there is a production of the reactive oxygen species. Initially, I mentioned because there is a blockage of this electron transport chain leads to accumulation of the electrons that can lead to production of the ATP, and also at the same time the production of the reactive oxygen species. Then what we thought is that probably the operation of the cycle can also reduce the reactive oxygen species. Therefore, what we have done, we checked the ROS levels by using DCF fluorescence and also NBT and also DAB, and we found that under anoxic condition there is increasing levels of the reactive oxygen species. and which got suppressed in the presence of the nitrite, clearly suggesting that operation of this phytoglobin NO cycle can lead to tight regulation of this cycle. Therefore, there won't be any electron leakage. Thereby, the superoxide production does not occur in the presence of the nitrite. And then further to elucidate how this cycle is helping in the reduction of the reactive oxygen species is that previously I have shown you that the nitric oxide is inducing alternative oxidase is a actually protein that is present in the mitochondrial electron transport chain. What we have done was we have actually checked this the expression of the alternative oxidase in the aerobic and anaerobic condition in the absence or presence of the nitrite. You can see the alternative oxidase levels have enhanced under low oxygen condition. At the same time, we checked the capacity of the alternative oxidase and cytochrome C oxidase. We found that in the presence of the nitrite, the plant switched from the cytochrome pathway, which is a light orange color, to the dark orange. You can see here you can see the switch from the cytochrome pathway to the alternative oxidase pathway. And then what we have done was to further confirm, we have grown these plants in the presence of alternative oxidase inhibitor. You can see the anoxia induced growth got inhibited in the presence of the salicylic hydroxymic acid, which is an inhibitor of the alternative oxidase. This is actually proving that alternative oxidase also induced under low oxygen condition along with the phytoglobin NO cycle that actually reduces the reactive oxygen species during the anaerobic germination. And then also the maintenance of the internal oxygen, low oxygen, there is oxygen homeostasis in the roots is very important, which can be also regulation of this oxygen is also very important in the roots in order to maintain the homeostasis. What we have made a setup where actually a micrometer the 10 micrometer this needle we coated with a phosphorescent dye, we injected into the roots and then checked the oxygen level. We found that in the presence of the nitric oxide, they maintained very good oxygen levels. And then finally, what we have done was we have actually did a survival experiment by actually growing these rice seedlings in the absence or the presence of the nitrate. What we have done, we have grown them. We have actually completely submerged these rice seedlings for 15 days, completely submerged. And after that, what we have done was we have actually reoxygenated. And you can see the plants which are grown in the presence of the nitrite survived finally, but the plants which are actually not treated with the nitrite not survived. This clearly suggesting that nitrite plays very important role in generation of the ATP and also driving the alternative oxidase pathway to actually minimize the reactive oxygen species during the anaerobic germination. So finally, this is the pathway we have elucidated that nitrite that is accumulated, nitrite that is also present in the soil also helps, which actually leads to induction of the nitric oxide by the cytochrome C oxidase B6 unit and also COX6B, and that leads to production of the nitric oxide. The produced nitric oxide actually drives the phytoglobin NO cycle here. This phytoglobin NO cycle leads to production of the NAD. At the same time, it also helps in production of the ATP. That actually leads to ATP production. At the same time, the nitric oxide that is produced can also induce the alternative oxidase that can actually reduce the reactive oxygen species. So finally, for the producing ATP, reducing reactive oxygen species, that helps in germination of the rice under low oxygen conditions. So these are our the different papers that we published in 23 and 24. And if nitric oxide pathway, if anybody is interested, you can read this nitric oxide, the regulation of the plant metabolism that we published in Molecular Plant. And then this paper that I have presented here, main paper, is phytoglobin nanocycle and AOX pathway play a role in the anaerobic germination, published in the Plant Cell Environment last year. So finally, I thank my collaborator. This work was mainly done by Aparajita Kumari and also Pooja Singh, Chandra Kaladhar, and Nidhi Yadav. So these people have contributed for this work. And I thank my several collaborators, the Lewis Mood, where we are working on the nitrogen use efficiency, and Professor Gary Lok. Unfortunately, Gary passed away last week. in very growing stage. And George Radcliffe, we work on the metabolic flux analysis. Dr. Shubhra, we are working on the plant pathogen interaction. Andre Igambardev and me elucidated together on the anaerobic ATP synthesis. Alistair Finney is one of our major collaborator on the metabolic analysis. And Christian Foyer, we are working together on reactive oxygen species and redox regulation. And Ashwini Parikh and Sneha Parikh, we are continuously collaborating on various aspects, including the role of nitric oxide in the salinity stress. Teresa Fitzpatrick, we are working on vitamin B6 pathway. So I thank especially the Department of Biotechnology, which is a major funding agency for providing funding and support from NIPGR and DBT, and several funding agencies like Indo-Swiss Grant, Indo-Portugal, Indo-German, and also Ignite Life Science Foundation recently funded our project on nitrogen use efficiency, etc. And I thank you so much for your attention. I am happy to take any questions.
Thank you. Thank you so much, sir. Sir.
Thank you, Dr. Jagdish. A very interesting and very exhaustive presentation on the role of nitric oxide and how it's synthesized, and then what all the key role it plays in terms of tolerance against hypoxia or anoxia stress. In this regard, I have a few comments.
Yes.
One is, we all know that it's nitric acid has a key role with regard to heavy metal tolerance, probably arsenic or cadmium.
Yes.
Does it also operate through the same pathway, or it's a different one?
That operates in a different pathway.
Okay.
So there are several reports that there is an activity which is nitric oxide synthase like activity that plays important role in certain the stressful condition like heavy metal stress, etc. And also recently there is a pathway that is elucidated. Recently there is a paper published in Molecular Plant that oxamine and hydroxylamine pathway. These are the two different pathways which are actually leads to production of the nitric oxide under aerobic condition, because this arsenic stress and all in the aerobic condition. But in rice, for example, rice is grown under low oxygen condition. Probably the nitrate reductase and mitochondrial electron transport chain can also play important role in production of nitric oxide in arsenic stress. But several reports says that the nitric oxide play actually important role in reducing the heavy metal stress via activating the antioxidant defense mechanisms. So this actually, so it's very interesting to check also the transition from the when rice is grown at different phases, how actually these pathways gets activated under normoxic condition and anoxic condition. What are the pathways? How these contribute for the heavy metal stress tolerance? That is an unexplored area.
Now talking about two extremes. One is upland rice, another is deep water rice.
Yes.
How do you expect the pathways? Will they be the same in upland rice, wherein it's grown under a lot of water stress, which means complete oxygen availability, and another one under deep water rice, wherein no oxygen is available, wherein you demonstrated very clearly
in deep water rice. We have also done some experiments even by using the IR64 and also MTU 1010. So what we found was actually...
No, these are not essentially upland varieties.
It's not upland varieties. So therefore, we have also checked the effect of zero oxygen condition anaerobic generation. And interestingly, we found that these rice varieties are also germinating much more better under low oxygen conditions, but not that much effective that you have seen the effect in the upland rice varieties.
And now a new method of cultivation called direct seeded rice.
Yes.
It could be wet direct seeded or dry direct seeded. It's coming up on one of the traits which is essential for rice line to survive under direct seeded rice, direct seeded condition is anaerobic germination. How we could exploit your discovery in terms of developing better breeding lines or varieties with better anaerobic germination?
Yes. So
are there any chemicals which we can add?
Actually, in our laboratory,
any seed priming agents we can add to enhance the germination under anaerobic conditions?
Yes. So in our laboratory, we have actually developed nanoparticles which can induce anoxic condition transiently. These are sodium diethionate nanoparticles coupled with the nitrite. Okay. So what they do is that they cause hypoxic condition as soon as you add. At the same time, they also release slowly and sustainable levels of the nitrite. Actually, we have done preliminary experiments by using these nanoparticles, and we found that when you treat this rice, even under aerobic condition, if you germinate them, these nanoparticles are inducing the germination. That is strikingly, this is a very, very important the results we got by using this nanoparticle. That means direct seeded rice, if you want to enhance the germination, but in the presence of the aerobic condition, by using this nanoparticle, you can actually transiently create hypoxic condition and induce these pathways to enhance the germination. And another approach is that by using genome editing. So ACPB is a protein that is actually present in the plasma membrane. Okay, there is a protein that is present, ACPB, that actually interacts with the ethylene responsive factors. Okay, so this ERF, ethylene responsive factors, are very important for induction of the alcohol dehydrogenase and also lactate dehydrogenase in order to increase the energy production. Okay, but this rice actually very much programmed to produce energy under low oxygen conditions. So the another approach is that to mutate this one ACPB protein. Therefore, the ERFs which actually the ACPB binds to the ERF, preventing the ERF to mobilize into the nucleus. So ERF can actually mobilize to the nucleus if you mutate it. That actually leads to more production of the energy that leads to enhanced germination. So these kind of approaches we can do. One is chemical approach by using also genome editing. One can check.
There are two things which are which can lead to better growth conditions under DSR. One is, of course, anaerobic germination and early seedling vigor. And I believe based on your lecture, nitric oxide, either endogenously synthesized or externally supplemented, can play a key role with regard to these things. And my colleague, Dr. Sridevi, she's working on field experiments related to DSR and weeds and all. And our head of department, Dr. Mahendra Kumar, unfortunately is not there here today. He has gone to Delhi to attend. You can get in touch with them if you want to experiment at field level any of the compounds which you have synthesized. Even under control conditions also, we have got biosafety glass houses, we can try it.
Okay, that would be great.
Because that will be one great service for direct seeded rice. What we anticipate is that in another 10 years, the total rice area, out of the total rice area, 30 to 50% may be under direct seeded rice. It has got certain advantages, as we have seen in COVID-19 times. It saves labor, it reduces the crop duration, and it also consumes lesser water. So if information that has been generated from your study can be applied for betterment of direct seeded rice, I think it's a win-win situation for everyone.
And another very important aspect is that one can also look into this the generation of the nitrogen oxides. For example, as I mentioned here, so when you grow this rice under flooded condition, there are several reports published in Nature Chemical Biology, Nature Climate Change, that rice, if you grow under flooded condition, there is an activation of the denitrifying bacteria that is present in the soil. That leads to production of the nitrogen oxides. This nitrogen oxides reacts with the ozone, then actually causes this NOx pollution, where we see the smog, etc. That actually leads to the reaction of the nitrogen oxides with the ozone. So another important aspect, if you want, we can collaborate on that. So in the difference in the nitrogen oxide and also this NOx pollution in the direct seeded rice and also the flooded rice, the comparison of these would definitely will definitely help in order to see how to actually whether direct seeded rice is helping to mitigate the NOx pollution as well. So that is another important aspect actually which can also add in.
Thank you very much. Now the house is open for any questions. First, I would like to invite our online participants. If you have any questions or queries to Dr. Jagdish, you can shoot off your questions. And following that, we can have our offline participants.
Dr. Suresh, he has raised hand.
Yeah.
Unmute
Suresh?
कर दीजिए.
Dr.
Dr.
Suresh,
Suresh, can you please ask your question in the chat box? Can you please type?
or you can no. Okay. As there are no questions from the online participants, we can now invite.
Can I
Yeah.
have some question?
Yeah.
Yes,
Go ahead.
yes.
Go ahead, sir. Go ahead, sir. Dr. Nagaraj. Yeah. Go ahead. Go ahead.
Got muted.
Or what I will suggest is online participants can type their questions in the chat box, which can be read out. So now we'll start the round of questions from our offline participants. Any questions? Yes. Mike.
Hello, sir. Myself, Satish Chavan, scientist nematology. First of all, congratulations for very nice work and presentation. Like we know that reactive oxygen species and nitric oxide, they are excellent signaling molecules. But as compared to the role of reactive oxygen species, the role of nitric oxide is a little less studied or documented. Because of one reason, it is highly unstable gaseous molecule, so it is equally difficult to measure nitric oxide. But you used chemiluminescence and DAF FMDA. These are highly specialized techniques. They require equipment. They are highly expensive. So can you suggest some like cost effective methods or biochemical based methods that we can use for nitric oxide measurement?
Yeah, for example, as you said, the chemiluminescence detection method is very expensive.
Yeah.
So one thing I will tell you that we have established a free radical measurement platform at NIPGR. So anybody would like to measure, then contact us, and we will be able to measure nitric oxide and give you the data. First of all, this is not a whatever that option that you are asking. Okay, but if anybody would like to measure under SARR infrastructure, we have established this facility, and we can measure all the free radicals, superoxide, reactive oxygen species, the internal oxygen, pH level, everything simultaneously by using the platform. So we have established in 1 or 2 months it will be open to everyone in India. Okay, you can measure it. So I am the coordinator of the program. And another aspect is that diaminofluorescent dye acetate, in most of the institute you have fluorescence microscopy, but the dye is a bit expensive.
Yes.
So in order to tackle that, we have actually standardized a method which is called as gas phase Grice reagent assay.
Yes.
Okay. So what you have to do is that there is a setup which I've actually described in one of the Journal of Experimental Botany methods paper. I will share you. So in this method, actually by using two agents, one is sulfalinamide and naphthalinamide, actually you can actually stimulate the nitric oxide and then oxidize it, and oxidized form of the nitric oxide can go to these two reagents that leads to production of the color, okay, which you can measure. That is a calorimetric method. Okay, so I can show you the methodology, but it's quite cost effective. I think you're going to several reactions in 1,000 rupees. So and measure the nitric oxide.
So these are nitrite and nitrate assays.
So
Grease reagents.
What you do is that you stimulate the nitric oxide. Nitric oxide is released in the form of gas. The gaseous form of the nitric oxide, you actually dissolve this gas into Grice reagent.
Oh, okay.
So that leads to, that means you are measuring only nitric oxide, which is actually oxidized.
Yeah, that is good,
Yes.
because this nitrite and nitrate assay, they are indirect measurement of nitric oxide. So
it's a gas-phase Grice reagent assay which require a small setup which can actually manually design. It's also good for students at all students and other people to design that one. So I will share you the setup.
Another one quick question regarding presentation. As we know that nitric oxide is produced in plants by two major pathways, nitric oxide synthase and nitrate reductase.
Yes.
Very nicely you shown that nitrate reductase is involved in this production of nitric oxide. Do you think nitric oxide synthase is also involved in this hypoxia condition? Or
No.
is
Nitric oxide synthase requires in animal systems, I can say, plant system, there is no nitric oxide synthesis so far discovered.
Huh.
But using L-arginine is a substrate that for nitric oxide synthesis, people see the production of the nitric oxide. So that is the only proof. Okay. So, but this, but it requires oxygen. Okay, it requires oxygen, nitric oxide synthesis. But recently, there is a paper published in Molecular Plant that there is another route which is called as oxamine. So you can go through it. It just published 2 weeks ago. So you can, that pathway can also produce nitric oxide under normal condition. So, but it just got discovered. Yeah, our spotlight on that also will come soon in Molecular Plant.
I have a couple of questions on role of nitric oxide in rice nematode interaction, but I will discuss this after this one.
But this is very less explored area.
Yes,
nematode interaction.
Yes,
Yeah.
that's what I will. Say. Sure.
And Satish recently completed his PhD from Wagner University.
Yeah.
Studying the interaction between nematodes and the crop plants.
I think there is a relevance that we have observed because I have a startup, Fruitech Private Limited, have established. In that, we have developed a novel formulation and to enhance the shelf life of fruits and vegetables. Okay, but in most of the cases, the fruits which are treated with our formula, as I think it's in, if you take custard apple and all, I think these are nematodes. I think inside. I don't know, but the levels of the insect infestation and everything has reduced in the fruits during the post-harvest storage. So that's what we observed. But direct effect of the nitric oxide and the nematodes is very unexplored area.
Yes,
It would be very, very interesting to explore. So,
yeah. Thank you, sir.
Yeah, I have a continuation of that. There are, yeah, it is on. I think I'm. Yeah, we have two interests in nematodes and this thing. In fact, first of all, I'll congratulate you for presenting such a complex subject in a crisp and straightforward. And it has taken me back to my nematode physiology class 30 years back. So actually, mostly nematodes are basically aerobic, and we have more nematode damage in the aerobic rice. But we have a specialist nematode, which is a rice root nematode, which is adapted to infect rice under flooded situation. Okay. And physiologically, it is adapted, like as you're telling, under anaerobic respiration, ADH and LDH.
Yes.
So there is, since accumulation of lactic acid is lethal to the nematode, like how we are getting cramps in case of athletes also. This nematode switches over to the ADH. And nematode has no separate respiratory system. It is cutically permeable. This alcohol is dissipated so that nematode is able to survive despite producing energy under anaerobic respiration without accumulation of lactic acid. It is very specialized nematode adapted, and we sometimes call as symbiont. Because it adapted to the rice. And when you are talking that microbes and the roots compete for oxygen, low oxygen level, I don't know what is the kind of significance of this nematode under the situation, whether it's putting more stress under anaerobic situation because it's adapted. That is one part to be explored. And the other one, Dr. Satish has recently completed nitric oxide as a signaling molecule for inducing resistance to the nematode. That he will be interacting with you on that. But this anaerobic situation, what is the interaction between rice root nematode roots in the field condition? Because what we have done is controlled experiments. So how they compete with each other, and is there any way to minimize their loss? And those are some of the areas I think to be explored.
So
and one more thing, like here also you have seen elevated lactic acid levels. Is it not like animals? Is it not lethal to the plant at that high levels of lactic acid produced?
So high level
if it gets accumulated in the system.
High level of lactic acid actually accumulation leads to acidosis. Cytosolic acidosis. Actually, that is actually a lot of proteins are pH sensitive. So the protein activity gets reduced. So it has to be balanced. So that is one thing. And another thing regarding your nematode interaction is that you said that there is a mutual, probably it is like a symbiont. So this is actually perfect time you have asked. In our platform, we have established established a technique to measure internal oxygen by scanning, there is a method called VisiSense, which is a scanning of the oxygen, pH, and several other parameters simultaneously. So I think that is a perfect platform for you to check how that computation is occurring between the nematode, how much oxygen at different development stages of this nematode is affecting the plant oxygen, whether there is any competition, etc. So this facility will be open in 1 or 2 months. If you can approach, then we will be able to actually help you to measure all the all these parameters, etc. And you can see, you can visualize with the eyes how the oxygen dynamics are changing. So in the presence of the nematode, whether there is any competition for oxygen, etc., under different conditions.
Yeah, that will be wonderful.
Yeah,
I think it
yeah,
will add.
sure.
Also,
Yeah.
like to suggest is that
Yeah.
at field we have got a nematode sick plot,
Okay.
and our team is also very competent to study the interaction between molecules and also nematodes and their effect on rice under in vitro conditions also.
Yeah.
What I would suggest is that your nano formulation, we can sign a MOA and then we can work together.
Sure.
One thing what I would like to see is whether it improves anaerobic germination under DSR conditions.
Yes.
Second, whether it improves the seedling vigor, which I expect based on the results what he presented.
Yeah.
Another one, whether that nano formulation which results in accumulation of nitrous oxide, will it also have any control over some of the nematodes which affect rice?
Yes, yes, fantastic. Yeah, sure. Definitely. Yeah.
Kindly get in touch
It's a
with the respective-
multifaceted, the role of this nano formulation on various ways too. Yeah, sure.
It's fine.
Yeah.
Yes.
Sir.
Take the mic, ma, because online participants can.
Yeah.
Sir, in general field conditions, I'm Subarna working as a plant breeder scientist here, sir.
Okay.
So I have at field conditions, generally we observe the flooding condition and also the salinity problem together. So how this nitrate will be helping during the flooding and also the salinity problem?
Yes, yes. So
that is
yeah,
farmers face the problem.
problem
together,
both
like
because you have to see water.
inland salinity and all.
Yeah.
So is it useful? Can we apply urea during this flooding condition, or these nitrates will be useful if we give the external applications to the plant?
So we have published a paper which is called as nitro-oxidative stress. Okay, so there is a nitro-oxidative stress comes because this nitric oxide reacts with react to oxygen species and cause a stress called nitro. This is a new kind of new stressful condition. So you can go through those papers. See this what we have actually observed was that presence of this alternative oxidase actually minimizes this nitro-oxidative stress under salinity condition. But for your question, combined effect of salinity and flooding, actually that is completely unexplored. So people have worked on flooding separately, salinity separately, but both of them combinedly nobody has worked. So therefore, but I'm sure that there is some relevance there because this is an unexplored area at the same time. So we found that these proteins, that is alternative oxidase, etc., which are helping in the flooding tolerance, also helping in mitigating nitro-oxidative stress. So under salinity stress. Okay. So you can go through our paper and it is completely unexplored area. So it will be good to explore using different substances like urea, nitrate, and see the actually it is actually helping to actually improving the plant growth. Yeah.
Sir,
I'd like to know what is the effect of temperature on this. Your
Okay.
deep water, you are
Yeah.
trying to deep water as well as DSR direct.
Yeah.
There is certainly a temperature variation.
Yeah.
So what is the effect of this?
So the effect of again I say that the effect of combined effect of, you mean the combined effect of the low oxygen and the temperature, again it is an unexplored area because nitric oxide, see the signaling started, the work started in I think near to 2000. Okay, so unlike the animal nitric oxide research, it started in 1970s, etc., but is actually started around 15 years ago. Okay, so it is people have started elucidating the role of NO in several stressful conditions, but combined stress actually people have less explored. There is a lot of scope, but one can what we have actually found that whenever there is a flooding, there is a less light passage to to the plants. The light gets, very less, and and also the it becomes dark inside. Okay, because when there is completely flooded and if there is a turbidity, the plants actually experience low oxygen condition, at the same time dark also. What we found in our case, actually whenever we expose low oxygen condition and also darkness, the plant produce more and more nitric oxide. Okay, so probably that why they produce is that probably to survive much more better when there is less light and also low oxygen. So probably, so there is a paper we published in Plant Journal, the nitric oxide emissions from tobacco system under different light and dark condition, and you can actually read it, and it is unexplored area. On the top of it, if there is a temperature effect, I think it will be more interesting.
Yeah, Dr. Jagadis, congrats for wonderful presentation. I am Satyendra. What I could understand from the presentation is that under anaerobic conditions, the more accumulation of nitric oxide helps in more production of ATP that helps in germination. So how does it influences photosynthesis? That is one question, like whether it has got any side or positive impact on photosynthetic machinery. And second part is how much is the environmental release when it at the cellular level, when the nitric oxide is formed and more nitric oxide is formed, how much is the release in environment to this process? Yeah.
So so we have checked the role of this nitric oxide and germination only. We haven't checked on later effects. Only the 15 day up to 15 days we have actually elucidated. But it would be interesting, I think. But whatever we observed is that so these leaves actually turn to be more greener in the presence of Yes, I agree. So probably actually photosynthesis is also very interesting to check during the early stages of growth of the plants by by using nitrate and all. This is again actually, as you said, that the respiratory pathway is also linked to the photosynthesis. There is more ATP, more reducing equivalent recycling, and that actual oxidative pentose phosphate pathway, and all these pathways also play important role directly or indirectly to the photosynthesis to generate this reducing equivalence. So it would be very interesting to see. So another thing is that how much is the release that we have actually already checked it. You can see the DAF fluorescence, diamino fluorescent diacetate, which can only measure nitric oxide which is internally produced, does not measure nitric oxide that is emitted. On the other hand, in another method, we have shown that chemiluminescence detection of method, which can only measure the emitted NO. Okay, but as you see the correlation, we cannot directly quantify both of them to match. As you see, in both cases, both methods actually leading to similar results, suggesting that probably whatever it is actually produced inside the tissue is also released to the outside environment. So this is the thing. But the thing is that the global impact, that means how much is produced, global impact, that we can actually with the smaller experiments, this can be scaled. Developing a small greenhouse where you can measure the completely in the closed environment, and you can actually translate that into per hectare how much is produced, and also per actually square kilometer how much is produced. You can
probably those estimates will help us to better understand.
Modeling can be done.
And one more question related to your nano nitric oxide component. So you said that it creates hypoxia environment, microenvironment.
Yes.
So if we apply in soil, what consequences it may have in other crops in the soil, which might have positive or negative?
So the low oxygen condition it transiently creates only in the span of we have measured these nanoparticles. Actually, nitrate is produced always, but the low oxygen condition is actually transiently produced over the period of 15 minutes. That's all. Actually, we found that we have created this nanoparticle for priming the plant defense responses against XAP, Xanthomonas oxinopotates per pomegranate. Okay, what we have done was these nanoparticles we sprayed on the plant that actually generated nitric oxide and also caused a hypersensitive response. Okay, and it mimics the hypersensitive response that leads to activation of the plant defense response. Okay, that's what we observed. And then we have actually, when we tested on rice, it was very, very interesting that under aerobic condition, when you treat, showed like anaerobic, the phenotype of anaerobic germination.
I think your intervention will have a lot of role, specifically I would say in legume crops, because there the water logging is the biggest challenge. Most of those legumes are highly sensitive for the water logging. So probably if you can test it in some legume models, and if it has some, I think it will be a kind of game changer.
Yes, like chickpea and all, when there is a unexpected or rains, there's a lot of damages occurring in winters. Yeah.
Sir, we'll take a couple of questions. Okay.
Congratulations, Gupta ji. You have given a good presentation. Just I want to know from you. You have tested for different genotypes also in between. Whether there is any interaction of genotype with the nitrogen oxide emission or this thing with different
genotypes?
Genotypes like MTU1010 or some other experiment.
Yeah.
With different varieties.
Yeah.
With other response.
Yeah, there is a difference actually between deep water rice and actually normal whatever the varieties that we are using.
So, from this we can able to identify some of the varieties which are suitable for these conditions.
Nitrogen.
Yes. We can as a screening method.
Yes. Screening can be done in larger scale, and to try to find out
can be screened.
Find out the correlation between these pathways and also the enhanced survival. Yes.
Thank you.
Madam, use the microphone. Benefit of online.
Dr. Jagdish, it's a very interesting presentation. I just want to know, is there any critical limit once hypoxia sets in the plant? For the external application of the nitrate, is there any critical limit or the time for its revival, or it will continue to go into another phase? Because there must be some difference in the nitric oxide upsurge and the application. So how do time it under field conditions? At least I'm talking about the application.
So that is very good question. That this has to be, it means we have to play with the system in the field condition. Most of the experiments we have done in the laboratory condition, but when it goes to the field, there are several factors which play important role. Even the other actually microbes which are present can also take up this nitrite. Also, they can also determine. So this how I think different concentration of formulation can be tested in the field, and also timely release of the particles. How does this affect on various parameters? Flooding tolerance, also germination can be also tested. So in field condition, it is much more complex, and that has to be identified for the application.
Sir, can we take up online questions? Only 3 questions are there. One question is, sir, the role of sugar signaling in anaerobic germination tolerance is established. So is there any congruence in nitric oxide and sugar signaling and hypoxia tolerance in rice?
Yeah, so the nitric oxide and sugar signaling and hypoxia tolerance, actually sugar signaling and hypoxia tolerance, that is correct. That is already known, but the nitric oxide relation with the sugar signaling is not known. There's a lot of this TOR signaling, TOR pathways are getting activated. That plays very important role. But one of our previous experiments we have shown that nitric oxide play important role in regulation of the carbohydrate consumption. Okay, we have done 14 carbon C labeling experiments where we actually we have provided to the seedlings and then checked the emission of 14 CO2. Okay, 14 this glucose is given, and then we have actually checked the 14 CO2 release, and we found that nitric oxide play important role in regulation of this carbohydrate consumption. And we have used the phytoglobin overexpression line, which produce less nitric oxide. When there is less nitric oxide, carbohydrate consumption has increased, which is also not good. So that means homeostasis of the carbohydrate consumption is also mediated by nitric oxide, but the deep signaling in relation between sugar, nitric oxide, and hypoxia tolerance is not elucidated much.
The example, what I would like to highlight is that one of the widely studied rice genes is sub1, and it induces quiescence or temporary dormancy.
Yeah.
So that the plants will not utilize the carbohydrate reserves and then grow under submerged condition.
Yeah.
But this gene is effective only for about 12 or 15 days.
Yeah.
So in this pathway, is there any role for say sub-1 mediated early stage submergence tolerance? Does nitric acid have any role?
No, we haven't checked that one. We haven't checked. We have actually played with the ethylene responsive factor, RAP 2.1, RAP 2.2, HRE1 and HRE2. I think this is one of the RAPs only, sub-1.
Yeah,
but specifically in rice, we have checked in Arabidopsis, but in rice, we haven't checked. So I think it's very interesting because how actually NO plays important role in actually via sub-1. So we are in touch with the Julia Belliceris regarding this Indo-US project, and we are taking up several genes in order to actually test them under different oxygen conditions, how these play important role. And Julia is very much interested in sub-1. Let's see anything comes. If anybody would like to collaborate.
There is one more set of genes called SNORKEL1 and SNORKEL2.
Sure.
They are diametrically opposite.
Yeah.
They offer late stage tolerance.
Yes.
Wherein the plant overcomes the quiescent phase and then grows rapidly.
Yeah.
So that under deep water conditions, the plant has the ability, as water level rises, it also knees and then rises beyond the level of water.
Stem elongation.
Stem elongation.
Yes.
Does nitric oxide have any role in it?
That is also not known. That is also, there are a lot of unexplored areas. Especially rice is very interesting system. Actually, it has actually furnished anaerobic germination. Another important thing is that it's actually always lying in the submergence condition. These pathways, how these are modulated, and also this stem elongation and hyponosty. And also recently there is also something leaf gas film formation. So under this condition, there is a gas film that is produced on the, for example, if you dip rice also, there is a film that helps in, whenever oxygen is required, plants use that, whatever the trapped oxygen. So there are a lot of interesting things.
Due to surface tension, am I right?
Surface tension, but the people are saying there is a plant adaptive responses. Actually, they found that when there is a scarcity of complete oxygen depletion, plants try to use this whatever this trapped oxygen that is in the gas films. So then they actually named it as one of the adaptive responses. So there are a lot of things can be explored in rice. So it is very interesting. Yeah.
Sir, we'll take up the next question quickly. Sir, nitrite role in mitochondrial protection is okay. How do you manage nitrite in soil during anaerobic events and vertisols to achieve this protection?
Yeah. So that already ma'am has actually asked the question. The management of the nitrite in the field condition is not yet tested. Actually, so that without collaboration, we can test the management of the nitrate. How does this affect the other microbial communities and also the tolerance to the flooding that can be actually explored. Yeah.
How stable can you maintain anoxia condition in your experiments, and the exogenous application of nitrite could influence that experimental setup, I guess. Could you please explain on it?
Yes. So how we manage the hypoxia experiments is that one is the bioxia chamber that we use. There is a glow box where we add nitrite, etc. But in large scale experiment where this, for example, 10 to 15 experiment which cannot be done in hypoxia chamber alone. In that condition, what we have done, we have actually designed polyacrylic boxes, which is a transparent, which has an inlet and outlet, which we have connected to the anoxic condition. At the same time, inside there is a technique that we use. It's called presence optic fiber. Actually, there is a sensor spot which has to be fixed inside the chamber, and from outside there is an optic fiber which can measure oxygen that is present inside the system. For example, if you want to measure the oxygen level inside this bottle without touching, without going inside, you have to fix this optic sensor spot on the top of the bottle, and then put the optic fiber there. Then it will tell how much oxygen present inside the system without going inside. Like that, we managed to control the low oxygen condition. At the same time, to add this nitrite, what we have done, we have covered a small hole with a paraffin, and then we have injected with a syringe this nitrite time to time for the treatments. So, and as soon as we inject and we close it, At the same time, we have a control of the optic fiber. If there is any change in the oxygen, we again pump the nitrogen gas. Thereby, we maintain the low oxygen condition.
Low oxygen conditions can also be maintained by pumping in carbon dioxide.
It can be maintained by carbon dioxide. People use the carbon dioxide also to maintain, but usually we prefer nitrogen gas.
Because it inert, no?
Yeah, right.
Okay,
Are
sir.
there any
Any
more
other
questions?
questions? Yes, it's done.
So we can go to the next item.
We'll felicitate, sir. After that, we'll go for felicitation. Yes. Sir, we please call you. Felicitation.
To honor our invited speakers. Thank you. It's also a certificate of appreciation from our institute. Thank you. Thank you.
Thank you all.
So we can sit here if you want, or
Yeah, please. You can sit here.
do you have any snacks or?
I request Dr. Padma Kumari ma'am to please propose a formal vote of thanks for the webinar.
Dr. Padma Kumari is one of the senior most principal scientists in entomology, and Madam is also our seminar committee in charge. Thank you.
Ma'am, can you just switch off the mic, please? Thank you.
Okay.
Yeah, done.
Am I audible?
Yes, yes.
It's my proud privilege to propose the vote of thanks. Now I thank Dr. Jagadis who has come forward to deliver this talk. It's a very interesting talk on nitric oxide signaling and its role in hypoxia tolerance in rice. I think the rice is the best crop, I think, where you can work out all the mechanisms because it grows in all the ecosystems and it has a host of insect pests and parasites also which survive in hypoxia conditions. So it is only an initiative. I think we can go much forward beyond this. And I profusely thank our director for his vision and support in organizing the webinars, which increase the horizon of the knowledge base of our scientists. I thank the team of AKMU headed by Dr. Sailaja and team Biotech headed by Dr. Neeraja for addressing all the logistics in conduct of this webinar. And I see many ICAR scientists from sister institutes and my own colleagues who have taken active participation in the deliberations and they are still there to listen to us. Thank you very much. I think Dr. Gangaya is there from IIMR. I thank all the scientists from PJTSU, members of the SARR, and cooperators from all the ECRIP centers, students, project staff, who have taken active part in the deliberations, and many other dignitaries who are with us today. I thank one and all for their participation and making this webinar a success. Thank you. Thank you.